chapter photosynthesis, I will not go through from the beginning, okay? Because the beginning of chapter photosynthesis is just for you to understand on the concept of autotrophs, heterotrophs. Itu orang tahu dah kan? And then the site of photosynthesis, which occurs in a leaf of plant, and within the leaf tissue you have cells, which we call it as mesophase cell. So that is the place where photosynthesis occurs because it has so many chloroplasts. So as you know, chloroplast is the organelle involved for photosynthesis. And you know that organelles is a double membrane organelles. And then inside the chloroplast, there are many thylakoids, which why is it in the thylakoid? There are so many, why is it in the chloroplast? There are so many thylakoid because it is to increase, okay, the surface area so that there will be more chlorophyll pigment, okay, can be embedded in the thylakoid. So lagi banyak thylakoid, lagi banyak membrane untuk chlorophyll pigment embedded into. Okay, so as you know, chlorophyll pigment is directly involved in photosynthesis, as the function of the chlorophyll pigment is to absorb light energy. Okay, so light energy is in the form of photons. Okay, so the photon will be absorbed by chlorophyll and will be used as energy to carry out photosynthesis. Okay, and then photosynthetic pigments in the nodes, it is divided into three, which is you have the chlorophyll A, chlorophyll B, and also the other one is carotenoids. Okay, so chlorophyll A is the most important photosynthetic pigment because it is the most abundant. Okay, so it is the main photosynthetic pigment and it is the most abundant. So that's why when you see trees, okay, they are green in color because due to the presence of chlorophyll A will not absorb green light. Okay, it will reflect and transmit green light. So that's why you will see leaves green in color. Okay, so that one you have to understand on the concept of light spectrum lah kan, visible light. Okay, so visible light consists of the 7 colors of the rainbow kan, which is violet until red. Okay, so for chlorophyll A, it will absorb most in the violet blue range of wavelength and also green light. Okay, so if we look at the slide, okay, here, okay, so I will explain more on starting from this slide lah, okay. So photosynthesis, so you have to know absorption spectrum and also action spectrum, okay. So kalau absorption spectrum, it shows you, it is a graph, okay, to show you the different wavelength of light that is absorbed by a pigment, okay. So this one you have to understand. This is soalan yang suka keluar dalam exam kan, konsep light absorption by pigments that is used for photosynthesis. Okay, so absorption spectrum. This is a graph for absorption spectrum. Okay, so you have chlorophyll A and also chlorophyll B. So sometimes it also incorporate graph for absorption by carotenoids. Okay, so if you look at the graph, for chlorophyll A, it absorbs most in the violet blue range. Okay, violet blue range and also red light. Okay, okay, so that is for chlorophyll A, and it reflects. Okay, or reflects and also transmits green light. Okay, so this is the range of wavelength that chlorophyll A will not absorb. Same goes to chlorophyll B. So chlorophyll B, it absorbs in the range of blue light here and also red orange here. Okay, and it will not absorb. Salah. Tadi yang ni chlorophyll A. Okay, up to here. For chlorophyll B it is here. Okay, so basically green light. Okay, green light for both pigment will not be absorbed. Okay, so for chlorophyll A and B absorb light mainly in the blue region, which is 420 to 492. Okay, this one you can also include. Mas dia tak jadi salah if you say violet blue juga. Okay, violet blue region. Okay, and also red region. So this suggests that blue and red light works best for photosynthesis. So if you look at this graph, it shows you the absorption spectrum by carotenoids also. So carotenoids and also chlorophyll B, it function as accessory pigments. Yeah, accessory pigments. So by having accessory pigments, the chloroplasts, okay, the photosynthetic pigment can absorb more of the light energy to broaden. Okay, you see here, to broaden the spectrum, okay, to increase the amount of energy that can be used for photosynthesis. That is the importance of having accessory pigments. So there is more pigment to absorb more light energy. Okay, it broadens the spectrum again, and also the function of accessory pigment is to absorb excessive light that would damage the plant, the chlorophyll. So as you know, plant, they are sessile organism, they cannot move. So when they are exposed to hot temperature, excessive light, so they need somehow a kind of protection, okay, to protect themselves from excessive exposure to light. So that is the importance also of having accessory pigment to absorb excessive light, to protect them from excessive exposure to sunlight. Okay. So if you look at the graph for carotenoids, okay, so carotenoids are pigment that is orange, okay, orange, yellow, or red. That if the plant, okay, contains carotenoids, they will appear in this color, okay, yellow, orange, or red light, okay, because this light will not be absorbed, okay, they will be reflected and also transmitted. Okay, so for carotenoids, they will absorb most in the range of blue and orange. Kat sini. This is the range of wavelength that the carotenoids will absorb, and this is the range of wavelength that it will reflect. Okay, and also transmit. Okay, kalau apa maksud reflect and transmit? Kalau ini daun, kan? So reflected, like is like this. Transmit, it passes through the leaf. Okay, so maksudnya kat atas awak boleh nampak dia warna hijau, warna hijau kalau dia chlorophyll lah. Kat bawah pun awak boleh nampak dia warna hijau juga. Okay. So so for action spectrum, what is action spectrum? It is a graph, okay, of to show you the effectiveness of different wavelengths of light for photosynthesis. So for action spectrum, it tells you the range of wavelength that is the most effective to carry out photosynthesis. So to get this graph, you measure the rate, okay, rate of photosynthesis. Okay, rate of photosynthesis. Such as, for example, the release of oxygen. Okay, so release of oxygen, as you know, oxygen is the byproduct of photosynthesis. Okay, so you can also measure the growth rate. Okay, growth rate of plant. Okay, so for example, if any of you have looked up in the your future in the MOOC activity, I've embedded experiment. Okay, I've embedded experiment from the YouTube. It shows you, okay, if you expose plant to different sources of light, different colors of light, green, for example, or red or blue, okay, which of the plant will grow the most? Okay, so is it plant that is exposed to green light, or is it plant that is exposed to blue light, or is it plant that is exposed to red light, for example, which shows the most growth? Okay, macam tu lah. So you do that experiment to see the growth of plant when it is exposed to different kind of color of light. Okay, So action spectrum shows the rate of photosynthesis at different wavelength of light. So to obtain the action spectrum, the rate of photosynthesis is measured. Okay, at each wavelength for leaf cells or tissue exposed to monochromatic light of one wavelength. Okay, you expose to green light, or you expose it to blue light, or to green light, to red light, for example, or to purple light. So which shows the most growth, okay? For example, to measure the rate of photosynthesis. So from the action spectrum, as you can see, so it shows that photosynthesis is the most efficient in the blue and red light region. Okay, so this is from the presence of chlorophyll A in this region, and presence of chlorophyll A and also B and also carotenoids. Okay, so this one is contributed in the green region here, is contributed also by carotenoids. See, okay, see, also contributed by the presence of carotenoids in the leaf. Okay, in this area, chlorophyll A and B. Okay, so this is the action spectrum. Okay. So okay, so now we are going to look at the details. Yeah.
Saya ada soalan pasal action spectrum tu boleh?
Ah.
Kenapa different wavelength of light affect the rate of photosynthesis? Like kenapa tak semua wavelength dia kasih sama je?
You have to understand the ability of each pigment lah. So it depends on the structure of the pigment. So if you look at earlier in the diagram, so it shows you in this slide, okay, so this is the structure of chlorophyll A and also chlorophyll B pigment combined together. They include in one diagram. Okay, so says here chlorophyll A initiate light dependent, so it has a methyl group. So chlorophyll A, actually it has a methyl group. Okay, so it appears bright green. And as for chlorophyll B, it has terminal carbonyl group here. Okay, so in the porphyrin rings. So this allows it to be to appear as yellow green. Okay, so this is depend on the structure of the photosynthetic pigments that allows it to absorb different colors of light. Faham? Okay.
Okay, faham.
Okay, it depends on the structure. So this is the equipment, okay? The equipment used to measure the ability of different pigment to absorb different wavelength of light. Spectrophotometer. Spectrophotometer is really a useful machine, okay, to measure different kinds of measurement, okay? Okay, so photosynthesis. So photosynthesis basically it consists of two stages. The first stage is light dependent reaction, and the second stage is dark reaction. Okay. Another name for dark reaction is Calvin cycle, or it can also be referred to as C3 pathway. Okay, so although it is called as dark reaction, the Calvin cycle or the dark reaction has to occur during daylight. Okay, bukan maksudnya nama dia dark reaction dia boleh juga berlaku pada waktu malam, tak. Okay, it has to occur during daylight because in order for Calvin cycle to occur, it requires the product of light reaction. Okay, light reaction will occur first. Okay, during light reaction, you have photosynthetic pigment that absorb the light energy and convert the light energy and form ATP and also another molecule which is NADPH. Kalau kita tengok chapter photosynthesis, kalau kita tengok chapter cellular respiration, kita ada NADH kan? Okay, so during photosynthesis we have NADPH. Okay, so these two molecules, okay, will carry high energy electrons. Okay, it carries high energy electrons, and this electron will be released from the molecule and transfer along the ETC to synthesize ATP molecule. Okay, itu kalau dalam itu kalau dalam cellular respiration, tapi kalau dalam photosynthesis, NADPH will function as reducing agent, and it also has high energy electron to be donated to molecules in the Calvin cycle. Okay, so Calvin cycle, it has to occur during daylight because it requires the product of light reaction. Okay, so the purpose of Calvin cycle is to produce sugar. Okay, sugar, glucose lah. Okay, so in order to produce glucose, Calvin cycle has to occur during daylight. So ini misconception adalah, okay, bila ada reaction, dia boleh juga berlaku pada waktu malam, tak ya? It has to occur during daylight. Okay, so so basically these are the two stages of photosynthesis. We have the light dependent reaction, and so this is the first stage. Second stage is carbon fixation. Carbon fixation is referring to Calvin cycle. Okay, Calvin cycle. So light dependent reaction is the photo part of photosynthesis. So it is the stage where light energy is absorbed. So if you look at the diagram, so here photons, light energy in the form of photons will be absorbed by the photosynthetic pigments that are embedded in the thylakoid membrane. Okay, so thylakoid membrane is the place where photosynthetic pigments are embedded. Okay, it is the photo part. Light energy is absorbed. Okay, so as for the carbon fixation, it is the synthesis part of photosynthesis. Why? As I mentioned, that it is the stage where sugar is produced. Okay. Okay, so the second difference for light dependent reaction, also carbon fixation, is that for light dependent reaction, it occurs in the thylakoid. Okay, obviously, as mentioned, it is the place where chlorophyll are embedded, or photosynthetic pigments are embedded, and then we have carbon fixation, it occurs in the stroma. Ini adalah poin penting yang awak kena ingat, itu je. Okay, so and then light dependent reaction, solar energy is converted into chemical energy. Okay, so photon light energy is converted into chemical energy. So these are the chemical energy produced. Okay, ATP and also NADPH. Okay, so the product will be used during the Calvin cycle lah. Okay, so as you can see, for Calvin cycle, use the chemical energy, which is ATP and also NADPH, to make sugar. Okay, so basically, Calvin cycle will produce a sugar, we call it as G3P, glyceraldehyde 3-phosphate, and from this, it will synthesize glucose or fructose, okay, or any other sugar molecule, and also the building block of cellulose, for example. Okay, so it produces carbohydrates. So it says here, for most plants, C3 plants, the carbon fixation reaction is also known as Calvin cycle or C3 pathway. Okay, so kenapa kita panggil dia sebagai C3 plant? Sebab apa? Semasa Calvin cycle, carbon dioxide, okay, will enter into Calvin cycle, okay, and then it will be fixed. Fix maksudnya incorporate. Maksudnya carbon dioxide ini akan bergabung dengan organic molecule yang dah ada dalam Calvin cycle, dalam stroma, and produce a 3 carbon sugar. Okay, sugar. So that's why kita namakan dia sebagai C3 pathway. It is because the first product of carbon fixation is a 3 carbon compound. Okay, so that's why we call it as C3 pathway, and the plant we call it as C3 plant. Okay, so kalau kat gambar ni tengok, so light reaction absorb light energy, photons, and then it produces NADPH and also ATP, and this NADPH and ATP are released into the stroma where Calvin cycle occur. The light reaction, it produces oxygen as the byproduct. Okay, in order to produce oxygen as the byproduct, it requires photo molecule to be split. Okay, we call it as photolysis. Photolysis. So Krebs cycle, it requires carbon dioxide, it produces sugar or carbohydrates. Okay. So kita tengok stage yang pertama, which is light dependent reactions. So the most important component for light dependent reaction is the photosystem. Okay, so there are two types of photosystem, photosystem 1 and also photosystem 2. Okay, so each of the photosystem, it has two major components, which is antenna complex and also reaction center. Yes, reaction center complex. So in the antenna complex, it is also known as light harvesting arrays ataupun light harvesting complex. Okay, light harvesting array or light harvesting complex pun boleh guna juga istilah tu, depending on the books that you refer to. Okay, so antenna complex consists of chlorophyll A, chlorophyll B and accessory pigments bound to protein. So kalau tengok dalam gambar ni adalah protein. Okay, protein where all the photosynthetic pigments are bound to and they are embedded in the thylakoid membrane. Okay, so bahagian ni kalau tengok, this is the region we call it as, yang saya merahkan ni, the antenna complex. Okay, so within the antenna complex, the green circle represents chlorophyll A, chlorophyll B, okay, and also carotenoids. Okay, so another component of the photosystem is the reaction center. So this is the reaction center. Okay, so in the reaction center, it consists of a pair of chlorophyll A. Kalau awak refer kepada buku Campbell, dipanggil a special pair. Okay, a special pair of chlorophyll A, and also it contains primary electron acceptor. Okay, which is a protein function as electron acceptor. Okay, so the function of chlorophyll A ni nanti kan, at the reaction center, it has a special pair of chlorophyll A. It depends on the photosystem. That the P680 and P700. Okay, P680, it is inside the photosystem 2. Okay, P700 inside photosystem 1. Okay, so these two chlorophyll, special pair of chlorophyll A, it will have electrons. Okay, a pair of electrons, and when these electrons receive photons, light energy, it will be energized. Okay, it will have high energy and then will be released from the chlorophyll A and then released to the primary electron acceptor, and then from there the electron will be passed along the electron transport chain, ETC. So when the electron are passed along ETC, the electron will release energy, and the energy is used to synthesize ATP. Okay, so this is chemiosmosis. Ingatlah, dalam proses fotosintesis dan juga dalam proses cellular respiration akan melibatkan penghasilan ATP melalui the transfer of electron and juga chemiosmosis. Okay, cuma dia punya location dia je yang berbeza. So reaction center inilah special pair of chlorophyll A and primary electron acceptor. Kena imagine sebenarnya dia dalam sini, dia kat sini. Okay, dia letak kat luar ni nak menunjukkan yang daripada chlorophyll A ni akan release highly energized electron. Okay, highly energized electron, so dia akan release with high energy. So dia nak tunjuk energi electron tu sangat tinggi dan dia akan diterima oleh primary electron acceptor. Okay, so photosystem ada dua lah. tadi. So photosystem 1 and also photosystem 2. So short form PS1 and also PS2. Bukan PlayStation tapi photosystem. Okay. So photosystem 1, so in the reaction center, dia punya reaction center dia ada P700. So it is a special pair of chlorophyll A. Why do we call it as P700? Because it absorbs light energy in the range of 700 nanometer. This one you have to include the unit, okay? 700 nanometer. So that is the absorption peak. Absorption peak of P700, okay, which is the chlorophyll A found in photosystem 1. As for photosystem 2, the chlorophyll A is called as P680. Why? Because it absorbs light energy in the range of 680 nanometer. So both light is in the range of red light lah. Kat situ. Okay. So okay. So routes for electron transport in the light dependent energy. So two possible route of electron flowing through the light reaction. So these electrons, okay, it is the electron of or comes from the electron of the P700 and also P680. Okay, as the result of both chlorophyll absorb light energy, so the electron will be energized. All right. So the flow of electron, there are two flow or routes. Okay, which is the first route is the non-cyclic electron transport. Okay, ataupun kita boleh panggil dia sebagai non-cyclic phosphorylation. Okay, so non-cyclic electron transport or non-cyclic photophosphorylation. So the other one is cyclic electron transport, same, or cyclic photophosphorylation. Okay, so for non-cyclic photophosphorylation, it involves photosystem 1 and 2. As for cyclic, it only involves photosystem 1. For non-cyclic, it produces both ATP and NADPH. That will be used for Kevin cycle. For cyclic, it only produce ATP molecule. So kalau tengok kat sini, for photosystem 1 and photosystem 2, okay, dalam non-cyclic electron transport, yang akan terlibat dulu adalah photosystem 2 dulu sebenarnya. Okay, so photosystem 2, dia punya chlorophyll A, which is P680, release electron and transfer along ATC, okay, along ATC, and then received by P700. So this is in the photosystem 2. This is photosystem 1. Okay. So kenapa bagi nama photosystem 2 dulu? Kenapa kita bagi nama photosystem 2? Padahal dia works awal lagi berbanding dengan photosystem 1 which works later kan. Sebab apa? Sebab photosystem 1 was discovered first, okay, by the scientists. Photosystem 1 was discovered first, then only photosystem 2 was discovered. Okay. Tapi dia photosystem 2 yang akan berfungsi dulu. Okay, so kita akan tengok for the non-cyclic electron transport. So here kita akan tengok yang photosystem 2 akan berfungsi dulu. It will be activated first. Okay, photosystem 2 is activated when a pigment molecule in an antenna complex of photosystem 2 absorb a photon. So a pigment molecule in the antenna complex in this region. Okay, in this region. Okay, so it absorbs photons. Okay, so the photon will be transferred from one accessory pigment to another accessory pigment to another accessory pigment until it reaches P680, direction center of photosystem 2. Okay, so the energy is transferred from one pigment to another within the internal complex until it reaches the reaction center. So the process of transferring the energy is called as resonance. So in the reaction center, the energy reaches a P680 molecule. So the energy excites an electron of P680. So when the electron of P680 is excited, so the electron will be released. Electron. And so P680 becomes P680 plus subscript, superscript. So P680 becomes oxidized, which is it become P680 plus. So maksudnya dia dah lose an electron. Okay, so the electron is then rise to a higher energy level. So the energized electron is accepted by a primary electron acceptor, which known as phyophytin. Okay, a highly modified chlorophyll molecule. Okay, so semua ni berlaku kat photosystem lah, photosystem 2, right? So so kita sambung. So the primary electron acceptor passes the activated electron along electron transport chain ETC until it is donated to P700 plus in the photosystem 1. So mesti dia dalam keadaan yang oxidized. Okay, P700 plus means that it is lacking an electron. So the electron has to be replaced. Okay, so replaced by receiving electron that originates from photosystem 2. Okay, so P700 plus becomes P700, which is reduced. Reduced maksudnya dia apa? Terima elektron. Okay, it accept electron that was coming from photosystem 2. So the ETC connects photosystem 2 to photosystem 1. So as the electron travels down the ETC, the electron loses energy. So sebab itulah kalau tengok kat gambar ni, dia daripada atas pergi ke bawah sebab daripada primary electron acceptor, the electron has very high energy. Okay, so it will be transferred from one electron carrier to another electron carrier to another electron carrier of ETC until it reaches P700 plus of photosystem 1. So bila dia receive, dia akan jadi reduced, become P700. So ini kalau tengok kat sini adalah component of electron transport chain. Dia ada a few electron carrier. Okay, dia punya electron carrier dia kita namakan dia sebagai yang mula ni saya, kalau tak silap saya adalah, nanti kita check balik, PQ, PC, kat sini CYT. Okay, PQ number stand for ubiquinone. Okay, ubiquinone. Ubiquinone. Yang ni adalah plastocyanin. PC adalah plastocyanin. CYT adalah cytochrome. Okay, so those are the electron carrier of ETC. Okay, and all these are embedded in the thylakoid membrane. Okay, so as the electron travels down the ETC, electron loses energy. So the energy is released the energy released is used to produce ATP during chemiosmosis. Okay, chemiosmosis kalau awak ingat dia melibatkan apa? Pergerakan hydrogen ion, okay, from one region to another region and then diffuse back through ATP synthesis. ATP synthesis synthesize ATP. Okay, nanti kita akan tengok dengan lagi detail proses chemiosmosis kejap lagi. Okay, so alright, so tadi, okay, so kalau kita tengok gambar ni, the electron that was received, okay, by P700, now it has very low energy. Okay, the electron that is received by P700 has really low energy. So how to re-energize the electron? By absorbing photon again. Okay, so the accessory pigment, okay, of photosystem 1 in the antenna complex will absorb energy, pass from one pigment to another pigment until it is received by P700. So the photon will excite the electron. So the electron will be released from P700 and received by primary electron acceptor. Okay, sama je proses dia. So the energy is transferred to reaction center and reaches P700. So the energy is the energy re-energized an electron of P700. So the energized electron is transferred to primary electron acceptor, which is also a special molecule of chlorophyll A. So P700 become P700 plus. Maksudnya dia dah kehilangan elektron, loses an electron. Maksudnya dia apa? Oxidized lah. Okay, oxidized. Okay, loses electron to the primary electron acceptor. If you look at the box here, okay, P700 replace the missing, P700 plus, okay, replace the missing electron by one donated from photosystem 2. Okay, so kita pick up daripada sini, primary electron acceptor. The energized electron is passed along ETC and also passed to ferredoxine. Okay, ferredoxine is also the electron carrier of electron transport chain. Okay, ferredoxine transfer the electron to NADP plus. Okay, catalyzed by the enzyme ferredoxine NADP plus reductase. Okay, so only single electron pass down the ETC, but two electrons are needed to reduce one molecule of NADP plus to become NADPH. Okay, requires two electrons. So it means that NADP plus will accept two electrons and one protons and reduce to NADPH. So the function of NADP plus to function as the terminal electron acceptor. So NADPH is reduced to the stroma. Kalau tengok kat sini, kena fahamkan kenapa NADP plus kena receive dua elektron dan satu hydrogen ion. Satu hydrogen ion ni from medium, okay, from the medium in the stroma. Okay, the stroma will have hydrogen ions, okay, and then will be accepted by NADP plus. NADP plus meaning that it is lacking an electron. So in order to become NADPH, it has to receive two electrons and also one hydrogen ion. Then only it can become NADPH. So it means that the two electron, one will be received by the plus sign of NADP plus, the other electron will be received by the proton. Okay. So dia akan masuk satu kat sini elektron tu, satu kat sini elektron. Faham? Boleh So that's why it requires two electrons. Okay, so ingat balik dekat photosystem ni tadi, photosystem tu tadi, okay. So P680 was missing an electron because electron dia dah dipaskan along ETC sampai dekat photosystem 1 dan receive sampai di receive by NADPH molecule. So mana nak ganti elektron yang P680 tu tadi dah hilang? So dengan melalui proses photolysis of water. Okay, so replacing the missing electron of P680 via photolysis of water. Photolysis. Photo means that it requires light. Lysis, split. Okay. So P680 become P680 plus when it loses an energized electron to the primary electron acceptor. P600 plus is a strong oxidizing agent. Nampak? Plus, it is lacking an electron. It is missing an electron. Okay. So in order to replace the missing electron, the molecule will pull electron away from an oxygen atom of water molecule. Okay, so a water molecule is split by an enzyme into two electrons, okay, two hydrogen ions and 1 oxygen atom. Okay. So kalau awak tulis dia punya formula adalah 1 water molecule will be split, okay, using light energy and also catalyzed by enzyme into 2 electrons, 2 hydrogen ions, and 1 oxygen atom. Okay, so you know that the byproduct of light reaction is 1 oxygen molecule. Ingat ya, kalau molecule is O2, so requires 2 water molecule to be split in order to release an oxygen molecule. Okay, so then only oxygen molecule is released as the byproduct of light reaction. Okay, so the electron are transferred one by one to the P680 plus, thus reducing it to P680. Okay, so dia dah receive electron daripada photolysis of water, dia jadilah balik P680. Okay, so the hydrogen ion are released into the thylakoid lumen. The two water molecule must be split to form 1 oxygen molecule which is released as byproduct. Ini penting untuk awak faham. The hydrogen ion from the photolysis of water is released into the thylakoid lumen. Tu awak kena ingat. Okay. The hydrogen ion, so tengok kat sini dalam chloroplast ada thylakoid kan. Dalam thylakoid ni, so ini thylakoid lumen ataupun kita juga boleh panggil dia sebagai thylakoid space. Kat luar ni kita ada stroma. Okay. So thylakoid lumen ni will have a very high concentration of hydrogen ion. Okay. So one of the factor that contributes to the high concentration of hydrogen ion in the thylakoid lumen is from the photolysis of water. So that is one contribution of why thylakoid lumen has very high concentration of hydrogen ion. Ingat tu. Okay. From the splitting of water molecule will cause thylakoid lumen have high concentration of hydrogen ion. Okay. So this is the whole diagram. Okay. Kalau tengok gambar ni, you all mesti boleh explain the transfer of electron starting from photosystem 2 along the ETC, okay, and then received by photosystem 1 and then released balik along another second set of ETC, which consists of pyridoxine and ATP plus, and then received lastly by an ATPH. So kalau tengok, the electron was from water molecule received by photosystem 1 P680, transfer along ETC, okay, to photosystem 1 to primary electron receptor to another second set of ETC and received by NADPH. So maksudnya kat sini elektron tu dah tak balik semula dah dekat P photosystem 2. So that's why kita panggil sebagai non-cyclic. Daripada satu arah je, one way flow of electron. Okay, one way flow of electron. Daripada air terus sampai received by NADPH. Okay, so this is released into the stroma for Calvin cycle, to feed the Calvin cycle. Sama juga dengan ATP, also released in the stroma, okay, to be used during Calvin cycle to synthesize sugar. Okay, so in the presence, it says here, in the presence of light, the non-cyclic electron transport involve the formation of ATP, which is coupled, okay, with one way flow of energized electron from water to NADP plus and from NADPH. So sebab kita namakan dia sebagai non-cyclic electron transport or non-cyclic photophosphorylation. Okay, photophosphorylation sebab apa? Photo, during this process, it requires light energy. Phosphorylation, why? Because the process produces ATP also. Okay, so the product of the product of light dependent reaction is ATP and also NADPH which is released into the stroma and required during Calvin cycle, ataupun kita panggil juga ni sebagai carbon fixation. Carbon fixation maksudnya carbon dioxide, the carbon of carbon dioxide is incorporated. Okay, is incorporated with an organic molecule which is part of the Calvin cycle to form another product, okay, which is a 3-carbon sugar. Carbon fixation, incorporate, okay, with RUBP lah. Saya bagi terus, okay, which is function as the carbon dioxide acceptor. Okay, RUBP is the molecule involved in the Calvin cycle process. Okay, so dia akan jadi nanti 3-carbon sugar. Okay, 3-carbon sugar, 3 carbon. Nanti proses, detail prosesnya kita tengok lah. Sekali lagi, so that's why kita panggil juga dia sebagai C3 pathway. Okay. So the cyclic electron transport, so cyclic, nama pun cyclic, so you should know that the electron will be transferred, okay, in a circle flow, okay. So it only involve photosystem 1. Ni awak kena ingat the pathway is cyclic. So maksudnya from photosystem 1 along ETC back to photosystem 1. Okay. So P700 absorb light energy and excite electron. Excited electron are transported through the electron transport chain and eventually return back to P700. Okay. So energy release is used for the synthesis of ATP only. Okay, it only produces ATP and no NADPH are produced and no oxygen is released. That is cyclic. Okay, cyclic electron transport or cyclic photophosphorylation. The transfer of electron along ETC back to photosystem 1 balik semula releases energy to synthesize ATP during chemiosmosis. Okay. So what is the significance of having cyclic electron transport? Okay, so it is to generate extra ATP, to generate more ATP molecule, okay, for the plant. So it generates extra ATP to power other ATP requiring processes in the chloroplast, help maintain the optimal ratio of ATP to NADPH required for carbon fixation or Calvin cycle. So ini untuk memastikan akan ada lebih banyak ATP molecule. Okay, sebab kalau nanti kita tengok masa Calvin cycle ni dia ada 3 phase. Okay, phase 1 is carbon fixation, phase 2 is reduction phase, phase 3 is regeneration phase. Okay, so during phase 2, okay, so during phase 2 dia akan menggunakan ATP and also NADPH, the same amount of ATP and also NADPH. If I'm not mistaken, 12 molecule of ATP and also 12 molecule of NADPH. During the regeneration phase, dia akan guna ATP. Okay, so it means that the Calvin cycle will require more ATP molecule. So in the regeneration phase, it requires another 6 ATP molecule. Okay, so as you can see, more ATP molecule is required during Calvin cycle. So the benefit of cyclic electron flow is to produce more ATP molecule for plant. Okay. So comparison between linear and cyclic electron flow. Linear is referring to non-cyclic non-cyclic electron transport. That is linear, one-way flow one-way flow from PS2 along ETC, PS1 and then received by NADPH molecule. Okay, so that's why dia juga boleh kita panggil sebagai linear electron transport. Okay. So for linear electron transport, source of electron is water. Okay, water undergo photolysis of water, release electrons and also hydrogen ion and also oxygen atom. Electrons to hydrogen ion and also oxygen atom. Okay, so source of electron adalah air. For cyclic electron transport tak ada source. Maksudnya daripada PS1 balik semula ke PS1. Okay, so electron cycle through the system. Okay, this a cycle lah. Okay, for linear electron transport is oxygen release, yes lah, from photolysis of water. Cyclic, no. Okay, for linear, terminal electron receptor is an ADP plus. Okay, so dia akan accept apa? 2 electron, 2 hydrogen ion to become an ADPH. Okay, so terminal electron receptor will be an ADP plus. For cyclic tak ada. Okay, tak ada terminal. Maksudnya memang tak ada lah, tak ada awal, tak ada akhir. Dia memang a circle. Okay, a cyclic process. PS1 balik semula ke PS1. And then for linear, form in which energy is temporarily captured, which is ATP lah. So linear produce ATP and also an ADPH. For cyclic produces ATP only. For linear, involve both photosystem 2 and also photosystem 1. Cyclic only involve photosystem 1. Okay. So process chemiosmosis. Okay, so chemiosmosis, tahu dia adalah proses untuk synthesize ATP. Okay, so chemiosmosis in photosynthesis involve 2 regions, which is the thylakoid lumen and also stroma. So as I said, thylakoid lumen will have high concentration of hydrogen ion. Stroma will have low concentration of hydrogen ion. So you have to remember. Okay, so it says here as electron move down ETC, energy is released and used to pump proton. Pump, ingat saya terangkan sebelum ni, pump adalah proses active transport. Okay, active transport maksudnya proses tu memerlukan tenaga, energi. Energi tu datang daripada mana? Datang daripada the flow of electron, electron along ETC. Okay, so itu proses pump. Pump ni adalah protein. Okay, protein transport protein lah. Okay, transport protein yang fungsi dia adalah untuk transport proton ataupun hydrogen ion from stroma into the thylakoid lumen. Stroma low concentration, thylakoid lumen high concentration, from low to high concentration. So it is active transport. Okay, forming a proton gradient across the thylakoid membrane. So once there is a high concentration of hydrogen ion in the thylakoid lumen, okay, so what happens next is that proton will diffuse back into the stroma through ATP synthase, and the ATP synthase catalyzes the photophosphorylation of ADP to ATP. Kalau untuk proses fotosintesis, penghasilan ATP tu kita tambah dia foto kat depan. Sebab apa? Dia melibatkan light energy lah. Okay. Tapi kalau proses cellular respiration, chemiosmosis, kita namakan dia apa? Ingat hari tu? Oxidative phosphorylation ataupun substrate level phosphorylation. Tapi kalau untuk photosynthesis, photophosphorylation. Okay. So in this diagram, it shows you basically how chemiosmosis occurs. Kalau tengok kat gambar rajah ni, so the grey structure here is the thylakoid. Okay. So here is the thylakoid membrane lah. You have the phospholipid bilayer that function as the thylakoid membrane. So inside you have the thylakoid space. Boleh guna dua-dua istilah, thylakoid space ataupun thylakoid lumen. So thylakoid space will have high concentration of hydrogen ion. Okay. So in the stroma, in this region, okay, will have low concentration of hydrogen ion. So tadi kan apa yang menyebabkan there is a high concentration of hydrogen ion kat telakoid lumen? Satu daripada photolysis of water. The other source that contributes to the high concentration of hydrogen ion in the telakoid space is the pumping. Okay, pump hydrogen ion from stroma to the telakoid space. Okay, so those are the two sources. Okay, these are the two sources that cause telakoid space to have a very high concentration of hydrogen ion. Okay, so kalau awak tengok kat sini gambar kan, so this is photosystem. Okay, kita start dengan apa? Photosystem 2 dulu. So photosystem 2, the accessory pigment absorb photon. Okay, and then the photon is transferred from one pigment to another pigment until it reaches P680. So electron is excited, received by primary electron receptor and transfer along ETC. Awak tengok kat sini kan. So awak tengok semua ni, the photosystem, the ETC, the ATP synthesis, all are embedded in the thylakoid membrane. Okay, so kalau tengok, so yang kat tiga purple structure here yang saya bulat ni, so those are electron carrier of electron transport chain. Kita ada PQ. PQ apa? Ubiquinon. Okay, yang besar tengah ni adalah cytochrome complex. Sekarang dia short form CYT, cytochrome. Okay, complex. PC adalah plastocyanin. Okay, so electron are transferred from one carrier to another carrier until received by photosystem 1, and then energized balik by absorption of photon, and then released to primary electron receptor here, and then transferred to ferredoxin. FD is ferredoxin, to the enzyme NADP plus reductase to catalyze the reaction. So the electron are received by NADP plus H plus and become NADP H. Okay, so this is produced in the stroma. Okay, in the stroma. Stroma is where Calvin cycle occur. Okay, so kalau tengok, the transfer of electron tu akan release energy. So this energy will be used to pump hydrogen ion from the stroma into the thylakoid space. Okay, yang arrow warna merah ni. Okay, when there is a very high concentration of hydrogen ion in the thylakoid space, hydrogen ion will diffuse back, okay, through the ATP synthesis, diffuse back into the stroma. So pergerakan tu adalah daripada high concentration to low concentration. Itu adalah proses apa? Passive transport. To be specific, dia adalah facilitated diffusion. Sebab apa? The movement of hydrogen ion is through diffusion using ATP synthesis. Okay, from high concentration to low concentration. So when hydrogen ion diffuse back into the stroma, it creates a type of energy, we call it as proton motive force. Okay, the proton motive force will cause the ATP synthesis to synthesize ATP molecule from ADP and also inorganic phosphate. Okay, sebab untuk for you to attach inorganic phosphate to ADP molecule, it requires energy. So the energy comes from the movement of hydrogen ion from high concentration to low concentration, which is the proton motive force. Pergerakan tu adalah sejenis potential energy, which is proton motive force. Okay, so that is basically chemiosmosis lah. Okay, so untuk synthesize one ATP molecule, it requires 4 hydrogen ion to diffuse through ATP synthesis. Okay, so itulah dia. Itu adalah chemiosmosis. So saya dah terang dah semualah. Alright. So comparison of chemiosmosis in chloroplasts and also mitochondria. So both organelle will undergo chemiosmosis to synthesize ATP molecule. Okay, both organelles generate ATP by chemiosmosis. So, but as for chloroplasts, it involves what? Light energy to be converted into chemical energy, which is ATP. But for mitochondria, chemical energy from food into ATP. Okay, so cellular respiration basically it breaks down what? Glucose kan? Glucose through a series of reaction kan? Glycolysis, link reaction, the citric acid cycle, and also oxidative phosphorylation until it produces ATP molecule. So another difference is for chloroplasts is the movement of hydrogen ion tu kan. So ruang dia berbeza lah. Kalau untuk chloroplast, hydrogen ion is pumped into the thylakoid space from the stroma. From stroma into thylakoid space. The process is pump, active transport. Okay. And then from the thylakoid space back into the stroma, which is the process is diffusion from high concentration to low concentration through ATP synthesis, which is it involves the production of ATP in the stroma. ATP is released into the stroma. But as for the mitochondria, obviously dia punya ruang dia berbeza lah. So mitochondria, dekat dalam mitochondria melibatkan apa? Intermembrane space dan juga matrix. So hydrogen ion pump into the intermembrane space. From mana? From matrix. Ingat balik So matrix, kena ingat dia ada low concentration of hydrogen ion, intermembrane space have high concentration of hydrogen ion. So the process is active transport from low to high. So intermembrane space, when you have a high concentration of hydrogen ion, diffuses back into the mitochondrial matrix through ATP synthesis. So ATP synthesis synthesize ATP in the matrix. Okay, so that is the similarity and differences between chemiosmosis that occurs in chloroplasts and also mitochondria. Ini similarity atas ni. Okay, similarity. Okay. So gambar yang untuk explain benda yang sama jugalah. The differences between chemiosmosis in mitochondria and chloroplasts. So the only concept that you have to understand is the difference in the concentration of hydrogen ion at different region of the organelle. Okay, so kalau mitochondria, region dia adalah matrix dan juga intermembrane space. Kalau dia adalah chloroplast, region dia adalah thylakoid space and also stroma. The light colored area is where hydrogen concentration is low. The darker region here is where concentration of hydrogen ion is high. So ingatlah yang tu. Okay, so pergerakan from low to high require energy. Energy datang daripada mana? Transfer of electron along ETC. Bila dia dah accumulate dekat the dense area, so dia akan diffuse back into the lower concentrated area of hydrogen ion through ATP synthesis. ATP synthesis synthesize ATP daripada ADP and also inorganic phosphate. Okay, so itu je. Awak kena ingat region. Okay, region yang mana concentration hydrogen ion tinggi, yang mana concentration hydrogen ion dia rendah. Okay, so konsep pergerakan tu apa? Active transport ke passive transport? Kalau aktif guna energi daripada elektron. Kalau pasif dia disebabkan oleh pergerakan daripada high ke low, proton motive force. Okay, from high, the movement of hydrogen ion from high concentration to low concentration along ATP synthesis. Okay, so now we are going to move on to the next stage, which is the dark reaction, okay, ataupun Calvin cycle. Calvin cycle, although we call it as dark, it has to occur during daylight because it requires the product of light reaction, which is ATP and also NADPH. Okay, so kenapa kita panggil dia dark? Sebab process Calvin cycle, it does not require light energy directly. Okay, it requires light energy indirectly from the light reaction that produces ATP and NADPH. Okay. Okay, so carbon fixation reaction, which is the Calvin cycle, occurs in the stroma of chloroplasts. Itu awak kena ingat. So involve reaction that fix. Why do we call it as carbon fixation? Daripada perkataan fix. Apa maksud fix? Incorporate. Carbon dioxide enters the Calvin cycle and incorporated into another organic molecule in the stroma that involved in the Calvin cycle. Okay. So as carbon enters into the Calvin cycle, it leaves the cycle as a sugar. We call it as glyceraldehyde 3-phosphate. Nama G3P ni dah familiar dalam proses cellular respiration. It is a 3-carbon sugar. Okay, so also known as C3 pathway because the product of initial carbon fixation is a 3-carbon compound. Okay, so plant that initially fix carbon this way are called as C3 plant. Okay, so that's why kita panggil dia sebagai C3 pathway. The first product of carbon fixation is a 3-carbon compound. Nama dia adalah 3-phosphoglycerate. Okay, glycerate, 3-phosphoglycerate ataupun phosphoglycerate ataupun PGA. Okay, so Calvin cycle is anabolic. It means what? It requires energy. Where does the energy come from? ATP. And also another molecule, NADPH, function as the reducing agent. It has high energy electron also to reduce compound in the Calvin cycle. Okay, so it's used in the formation of organic molecule sugar from carbon dioxide. So it is divided into 3 phases. Carbon dioxide uptake. Kalau baca dalam buku Campbell, ini daripada buku Solomon yang awak ada tu. Tapi dalam buku Campbell dia juga guna istilah ni sebagai carbon fixation phase. Okay, sama je. And then phase 2 is carbon reduction phase ataupun reduction phase pun boleh juga. And then the third one is RUBP regeneration phase. RUBP is ribulose biphosphate, a carbon dioxide acceptor. It functions as carbon dioxide acceptor. Okay, RUBP is a 5-carbon sugar. So phase 1, carbon dioxide uptake or carbon fixation. So carbon dioxide will enter into the Calvin cycle, which occurs in the stroma, and combine with RUBP. It reacts with, reacts with RUBP, ribulose biphosphate. It is a phosphorylated 5-carbon sugar P. Dia ada phosphate dalam compound tu. Okay. So the reaction is catalyzed by enzyme ribulose biphosphate carboxylase or oxygenase, or short form as Rubisco. Okay, so kalau tengok kat nama ni, dia ada slash oxygenase, ribulose biphosphate carboxylase or oxygenase. Maksudnya dia punya substrate kat sini boleh jadi carbon dioxide dan juga boleh jadi oksigen. Okay, so ini adalah salah satu kekurangan Rubisco yang kita akan tengok nanti. Kalau cuaca panas, bila cuaca panas, RUBP ni, enzim Rubisco ni akan prefer bind kepada oksigen instead of carbon dioxide. Itu bila cuaca panas. So ini kita akan tengok nanti untuk adaptation C4 dengan chem plant. Okay. So kita sambung semula. So the reaction produce an unstable 6-carbon intermediate breaks down into 2 molecule of 3 carbon, which is phosphoglycerate or PGA. Okay, so the carbon of carbon dioxide has been fixed or incorporated into a pre-existing carbon skeleton. Pre-existing carbon skeleton yang RUBP tadi lah. Okay, fixed. So that's why it is also known as carbon fixation. Carbon is incorporated to another organic molecule. Okay. So kalau kita tengok gambar-gambar ni, awak kena fahamkan purpose carbon cycle adalah untuk hasilkan sugar. Sugar usually yang kita belajar adalah glucose. Okay, which is a 6-carbon sugar. Okay. Untuk menghasilkan glukos ni, 6 carbon sugar, awak kena terus fahamkan, okay, yang sebenarnya Kevin cycle ni dia akan berlaku dan dia akan memerlukan 6 molecule of carbon dioxide. Sebab dia nak hasil, purpose dia adalah untuk menghasilkan 1 molecule of glucose. Okay, so 6 molecule of carbon dioxide will enter the Kevin cycle. Kalau awak baca buku Campbell, Kevin cycle dia sebenarnya kalau untuk menghasilkan glukos, dia akan berlaku 6 kali. 6 cycle that occurs simultaneously. Dia berlaku serentak memfixkan carbon dioxide tu, 6 molecule of carbon dioxide. Okay, itu je konsep dia. Tapi nak senang, awak just faham kan, okay, Kevin cycle to produce 1 glucose requires 6 carbon from 6 molecule of carbon dioxide. So dia akan combine dengan RUBP. Okay, 6 molecule. So the reaction is catalyzed by the enzyme Rubisco. Okay, and form 6 molecule of unstable 6 carbon intermediate. Kenapa kat sini dia adalah molekul yang unstable? Sebab, okay, awak tengok bermula kat sini. Yang the kelabu circle tu adalah dia represent carbon atom. So 1 carbon atom from carbon dioxide combined with 5 carbon from RUBP. Okay, so dia akan form a 6 carbon compound. Okay, a 6 carbon compound. Molekul RUBP ni dia dah ada 2 fosfat. Okay, kita belajar any molecule that has phosphate group attached to it is a highly energized molecule. Bila besarkan dia lagi, kan, dia jadi 6 carbon with 2 phosphate attached to it, it causes the molecule to be very unstable and immediately the 6 carbon intermediate will split into 2 molecule. Okay, dia akan split. So split into 2 molecule of 3 carbon sugar. Okay, so maksudnya 6, kena kalikan dengan dia dengan 2 lah. 6 molekul, dah pecahkan dia 2. So you get 12 molecule of PGA, phosphoglycerate. 12 molekul PGA akan terhasil masa phase 1. Okay, 12 molekul. So fahamkan daripada 6 jadi 12. Sebab dia 6, dia split 2, so 6 kali 2, 12. 12 molekul PGA yang akan hasil. PGA is a 3 carbon sugar. So ini adalah first product of carbon fixation. So that's why pathway ni kita namakan dia sebagai C3 pathway. Okay, usually carried out by C3 plant. Okay. Okay, so kita enter kepada phase yang kedua, which is carbon reduction phase. Okay, second phase. Okay, so kita continue dengan 12 molecule tadi. 12 molecules of PGA is phosphorylated by 12 molecule of ATP. So maksudnya setiap molekul PGA ni dia kena receive 1 phosphate daripada ATP molecule. ATP release a phosphate, so dia akan release phosphate dan dia akan jadi ADP. Okay, so tengok PGA ni dia juga dikenali sebagai 3-phosphoglycerate. Maksudnya karbon ni karbon pertama, karbon kedua, karbon ketiga. Karbon ketiga dia ada phosphate attached. Okay, so by receiving another phosphate from ATP, dia akan jadi molekul ni lah. Kita namakan dia sebagai one, 3 bis phosphoglycerate. Nampak? Phosphate attached to carbon number 1 and carbon number 3. So the molecule is 1,3 bis phosphoglycerate. Okay, So 1,3-bisphosphoglycerate will be reduced by NADPH. Berapa molekul yang diperlukan? 12 lah. Sebab ada 12 molekul 1,3-bisphosphoglycerate. So when the molecule is reduced, it also remove the phosphate group, okay, and become another molecule which is glyceraldehyde 3-phosphate. Yang ni yang you nak, okay? Yang ini, this is the most important molecule that is produced during Calvin cycle. Okay, nampak? So dia akan remove phosphate. So phosphate of carbon number 1. So dia akan jadi glyceraldehyde 3-phosphate. Berapa molekul? 12. Okay. So from the 12 molecule of G3P, 2 G3P will leave the cycle, okay, to form 1 molecule of glucose. G3P, 1 G3P, it is a 3 carbon sugar. So dia perlukan berapa? 2 lah, 2 G3P molecule to produce 1 molecule of glucose. Okay, so nampak ada 6 carbon lah dalam glucose. Faham So bila 2 dah keluar daripada cycle, so akan remain 10 in the cycle, 10 remain in the cycle. So the 10 G3P will enter into the third phase. So what is the third phase? The third phase is the RUBP regeneration phase. Tadi phase yang pertama dah guna dah RUBP. So because it is a cycle, you have to regenerate back the carbon dioxide acceptor, the RUBP molecule. So nak kena generate daripada 10 G3P yang terhasil tadi. Okay. So 10 G3P molecule which will total up to 30 carbon atoms. Faham tak kat sini? 30. Setiap G3P is a 3 carbon sugar. 3 kali dengan 10. So total carbon yang ada dalam cycle Calvin cycle masa phase yang ketiga ni adalah 30 carbon atoms. Okay, from the 10 G3P molecule. So they will be undergo this process which is rearrangement. Okay, rearrangement to form 6 molecule of ribulose phosphate. Ribulose phosphate is a 5 carbon sugar. So daripada 10 G3P akan undergo rearrangement to form 6 molecule of 5 carbon sugar, ribulose phosphate. So the 6RP, ribulose phosphate ni, dia akan receive phosphate daripada ATP molecule. Berapa ATP yang diperlukan? 6. Okay, so ATP release phosphate, okay, release phosphate, dia akan jadi ADP. So dia akan produce apa? 6 molecule of RUBP, ribulose biphosphate. Ada 2 phosphate attached to the molecule. Okay, so RUBP is regenerated and the new cycle begin. Okay, so this is the whole diagram of Calvin cycle lah. So phase 1, carbon dioxide uptake. Okay, phase 2, carbon reduction phase. Phase 3 adalah RUBP. Tadi kita tengok yang untuk cyclic phosphorylation, dia punya benefit adalah untuk produce extra ATP kan. So one of the benefit for producing extra ATP is to supply more ATP for the Calvin cycle. Kalau awak tengok kat sini, ATP 12 digunakan masa carbon reduction. Another 6 is used during RUBP regeneration. Okay, so for Calvin cycle, a total of 18 ATP is used, okay, and a total of 12 NADPH is used to synthesize 1 molecule of glucose ataupun 2 G3P molecule. Okay, so maksudnya ini awak kena ingat juga. Ini 12 ATP is used and also NADPH is used 12 during the Calvin cycle to synthesize 1 molecule of glucose daripada 2 G3P yang exit during the carbon reduction phase. Okay, so that is Calvin cycle. Okay, so summary of the Kevin cycle. So Kevin cycle requires the product of light dependent reaction and ATP and also NADPH. For every 6 molecule of carbon dioxide enter the cycle, 6 carbon leave the cycle as 2 molecule of G3P. Ini kena ingat So maksudnya remaining 10. 10 akan remain in the cycle to regenerate back the RUBP molecule. Okay, so the 2 molecule of G3P produce 1 molecule of glucose or other sugars such as fructose, and the plant also uses glucose to produce starch, which is stored carbohydrate in plant, and also cellulose, which is it forms the cell wall of plant. Okay, so you can glucose and fructose can join to form sucrose. Dan sucrose adalah sugar yang paling banyak ada dalam buah lah kan. So buah. So sucrose can be harvested from sugar cane, sugar beet, maple sap to get the maple syrup. Okay, so sugar cane awak tahu kan? So sugar cane adalah sumber awak punya table sugar. Sugar beet juga. Okay, sugar beet juga. Maple syrup yang letak atas pancake tu lah. Alright. Selalunya. Okay. So kita break sekejap untuk 2 minit. Ada soalan? Faham ke dah lost somewhere?
Okay, ada. Masih boleh.
Okay. Lihat sekejap, 2 minit. Okay, so kita sambung dengan the next subtopic which is C3, C4 and chem plant. Okay, so C3 plant is the kebanyakan tumbuhan yang kita nampak ni adalah C3 plant. Okay, tapi kalau C4 dengan chem plant adalah tumbuhan yang duduk kat tempat yang sangat ekstrim, panas, cactus kan. Tapi kalau C4 ni macam contohnya dia boleh survive kat tempat yang agak panas, macam contohnya tebu kan. So itu adalah contoh untuk C4 plant. Okay. So kita akan tengok untuk yang seterusnya adalah photorespiration. So what is photorespiration? So for plant that is exposed to hot temperature, to hot areas, okay, it causes this plant to close their stomata. Kalau untuk C4 plant, dia akan close stomata partially. Partial closure of stomata. So what is the purpose of closing the stomata? It is to conserve water. Sebab pada harian panas terik, tumbuhan ni adalah tumbuhan yang static, sesal. Dia tak boleh berlari kalau cuaca panas, dia tak boleh lari cari tempat yang teduh kan. So dia punya mechanism adalah close stomata to reduce water loss. Okay, to conserve water through that is lost through transpiration. Okay, But bila dia buat tu untuk conserve air, dia akan menyebabkan proses fotosintesis tu terganggu. Okay, sebab apa? Bila close stomata or close stomata partially, it prevents carbon dioxide from entering the leaf. So concentration of carbon dioxide in the leaf will be very low. Okay, when the concentration of carbon dioxide in the leaf is very low, itu yang tadi saya mention pasal Rubisco. Rubisco, salah satu kelemahan Rubisco dalam Calvin cycle ni adalah bila concentration of carbon dioxide ni rendah, dia akan prefer bind kepada oksigen. Sebab apa? Concentration of oxygen dalam leaf akan meningkat sebab oxygen is the byproduct of light reaction. So when the stomata is closed, oxygen accumulate in the leaf. Okay, so when oxygen accumulate in the leaf, Rubisco will prefer bind to oxygen instead of carbon dioxide. When Rubisco cannot bind to carbon dioxide, Calvin cycle cannot occur efficiently and less sugar will be formed. Okay, less sugar will be synthesized. So reduces photosynthetic yield. Alright? Itu yang berlaku semasa hari yang panas. So, closing the stomata reduces access to carbon dioxide. Carbon dioxide concentration begins to decrease within the leaf, causes oxygen to build up because oxygen is the byproduct of light reaction. Okay, so this condition favors a seemingly wasteful process which is called as photorespiration. It occurs during hot dry day, okay, where photosynthetic yield is really low. Okay. So kalau tengok kat gambar ni, alright. So you have two pictures here, two diagram. So during hot day, okay, if the stomata is open, water is lost through transpiration. In order to preserve water in the leaf or to conserve water, stomata will close. When the stomata close, carbon dioxide cannot enter, oxygen cannot leave. When carbon dioxide cannot enter, Rubisco will bind to oxygen. Okay. So in most plant, okay, C3 examples of C3 plant. You have the soybean, you have the potato, rice, oats. Okay, so those are example of C3 plant that you have to remember. Okay, so ingat ni je. Tak payah nak bagi contoh-contoh lain. Okay. Soalan ni pernah keluar dalam exam batch dulu, alright? To give example of C3 plant. So in most plant, most plant are C3 plant, the initial fixation of carbon dioxide via Rubisco form a 3-carbon compound which is the phosphoglycerate PGA itu tadi. Okay. Photorespiration occurs when concentration of carbon dioxide is low in photosynthetic yield. Carbon dioxide cannot enter into the leaf, okay, because the stomata is now closed or closed partially. So in photorespiration, Rubisco fixes oxygen. Fixes mean Rubisco incorporate oxygen instead of carbon dioxide during the Calvin cycle. So the product split into a 2-carbon compound which is glycolate. Okay, glycolate. So the glycolate will leave the chloroplast and the glycolate will be oxidized in the peroxisome and metabolize to carbon dioxide in the mitochondria. Alright, itu proses yang berlaku semasa photorespiration lah. Okay, so photorespiration basically occurs during daylight. Sebab apa? Masa tu hari panas terik. Okay. Requires oxygen and organic fuel. Produces carbon dioxide and water. Does not produce any ATP. Does not produce sugar or reduce photosynthesis efficiency, reduces photosynthetic yield. Dia produces less sugar or no sugar. So flowering plant have evolved 2 different mechanisms to avoid photorespiration. So one mechanism is for C4 pathway, the other is CAM pathway, crassulaceous acid metabolism. So ini sebenarnya dua-dua ni adalah adaptation. Okay, adaptation by plants that live in hot area or when they are exposed to hot conditions. Okay, C4 and crassulaceous acid metabolism. Dia nak prevent dia nak overcome photorespiration. Okay, an adaptation of plant to overcome photorespiration, C4 and also CAM pathway. Okay, so kita tengok for C4 dulu. C4 plants. Okay, C4 plants, they live in hot and dry environment. So example, you have the maize plant. Maize is corn. Okay, sorghum, millet, and also sugarcane. So these are example of plant that can survive in hot temperature. Okay, so they have adaptation, which is C4 pathway, to reduce photorespiration by incorporating carbon dioxide into four carbon compounds. Okay, which is oxaloacetate. Oxaloacetate is a 4 carbon compound, and it is the first product of carbon fixation. So that's why we call this plant as C4 plant. The first product of carbon fixation is a 4 carbon compound, oxaloacetate. I will short form it as OAA lah. Okay, as I explain, okay, throughout the notes yang ni. Untuk C4 plant sebenarnya dia akan undergo carbon C4 and also CAM applied also carbon fixation twice. Carbon fixation twice. Ingat tu, carbon fixation twice. Yang pertama adalah yang melibatkan carbon dioxide combine dengan PEP. So this function as the first carbon dioxide acceptor. First carbon dioxide acceptor. The second carbon fixation occurs during Calvin cycle. Okay, Calvin cycle, which is dia melibatkan carbon dioxide combine dengan RUBP to form PGA. Yang ni carbon dioxide combine dengan PEP akan form oxaloacetate. Ingat C4 dengan plant undergo carbon fixation twice. C3 carbon fixation only once. Okay. Okay, so for C4 plant, so dia ada dekat dia punya leaf akan ada dua jenis cell. Okay, mesophyll cell and also bundle sheath cell. Okay, remember how to spell sheath correctly. Okay, sheath, bundle sheath cell. So mesophyll cell is the main photosynthetic cell lah. Okay, so mesophyll cell are loosely packed between the bundle sheath cell and the leaf surface. It is the site of C4 pathway. So mesophyll cell is the site where C4 pathway occur. The first carbon fixation occurs in the mesophyll cell. The bundle sheath cell is where the second carbon fixation occur. Basically, it is the site of the Calvin cycle. So sebab dia adalah site untuk Calvin cycle, awak tahu Calvin cycle untuk produce sugar kan? Okay, so sebab dia Calvin cycle menghasilkan sugar, so that's why awak tengok dia punya susunan sel untuk leaf anatomy ni, bundle sheath cell akan surround ataupun near the vein. Okay, so when the Calvin cycle produces sugar, the sugar will enter into the vein, alright, and then transported to other part of the plant to be stored in other part of the plant, such as the leaf, the fruits or the roots or the tubers of the plant, okay, to be stored. So ingat, mesophyll dengan bundle sheath cell for C4 plant. So kalau saya akan explain daripada gambar ni je untuk C4 pathway, okay. You look at the diagram. Just look at the diagram. Okay. So the first carbon fixation occurs in the mesophyll cell. Okay. So first carbon fixation involves carbon dioxide to enter into the cell. Okay. But remember, the concentration of carbon dioxide is very low because stomata closed or closed partially. So carbon dioxide, although the concentration is really low, it can still undergo carbon fixation. Okay, but the first carbon fixation is by combining carbon dioxide with phosphoenopyruvate ataupun short form ni adalah PEP. Phosphoenopyruvate will function as the first carbon dioxide acceptor. Okay, acceptor. Okay, so PEP, phosphoenolpyruvate, which is functional as a carbon dioxide acceptor, combined with carbon dioxide, catalyzed by the enzyme PEP carboxylase to form oxaloacetate, OAA, which is a 4-carbon compound. What is the benefit of this first carbon fixation? Benefit of this reaction is that the enzyme PEP carboxylase will still bind to the low concentration of carbon dioxide dalam leaf tu. Walaupun carbon dioxide rendah, dengan adanya reaction ni, dengan melibatkan PEP carboxylase, PEP carboxylase akan tetap bind kepada carbon dioxide untuk menghasilkan oxaloacetate. Imagine kalau tak ada stage ni, dia terus proceed ke Kevin cycle, proses photosynthesis akan turn kepada photorespiration sebab dia akan terus melibatkan rubisco yang akan bind kepada oksigen instead of carbon dioxide. Itu pentingnya C4 pathway ni sebab dia melibatkan PEP carboxylase, enzim yang akan bind kepada carbon dioxide walaupun carbon dioxide concentration is really low. Okay, so oxaloacetate will then converted into malate. Malate is also a 4-carbon compound. All these reactions still occurs in the mesophyll cell. But once malate is produced, malate will enter into the bundle shaped cell through plasmodesmata. Ingat chapter 3, ingat plasmodesmata, alright? So malate enters into the bundle shaped cell through the plasmodesmata. Malate will split into 2 different molecules. The first molecule is carbon dioxide. Carbon dioxide is 1 carbon, right? Okay. Pyruvate is a 3-carbon compound. Okay, so dia akan split, satu jadi carbon dioxide, satu jadi pyruvate. So kita tengok untuk pyruvate dulu. So pyruvate enters back into the mesophyll cell and regenerate back the PEP. Okay, to continue the reaction lah. Alright, back to carbon dioxide. So carbon dioxide that is released in the bundle sheath cell, okay, will accumulate in the bundle sheath cell and causes carbon dioxide concentration to increase. And then only carbon dioxide can enter into the normal Kevin cycle to produce sugar. So the sugar, when it is produced, enter into the vein to be transported to other parts of the plant. Okay, so basically C4, ingat ini adalah misinterpretation kebanyakan student. Okay, kalau C4, kalau soalan, adakah does C4 requires rubisco? It still requires rubisco. It still requires RUBP because for C4 plant, it still undergo the normal Kevin cycle to produce sugar, but the location is different. Okay, the location for Kevin cycle is in the bundle sheath cell. The location of the first carbon fixation is in the mesophyll cell. The second, tadi saya cakap carbon fixation occurs twice kan? So maksudnya second carbon fixation occurs during the Kevin cycle. Okay, first carbon fixation in the mesophyll cell, second carbon fixation in the bundle sheath cell that involve the Kevin cycle itself to produce sugar. Okay. Selalunya kalau yang awak tengok molekul ATP and ADPH ni selalunya jarang ditanya lah. Tapi dia terlibat juga dalam reactions untuk C4 pathway ni. Okay. So this I've explained all. Okay. So this table it shows you the difference. Okay, the difference between C3 and C4. Good. Kalau C3, most plant are C3 plant, rice, tobacco, wheat, oat. But C4 plant, so these are example. Corn, sugar, sorghum. Okay. So example of C4. For C3 plant, carbon dioxide acceptor is a 5-carbon compound, RUBP. So this is during the Calvin cycle. Okay. For C4 plant, the first carbon dioxide acceptor is PEP, phosphoenolpyruvate in the mesophyll cell. Okay, the second carbon dioxide acceptor is RUBP itself in the bundle sheath cell, which is actually involve the Calvin cycle. Okay. So kalau untuk C3 plant, carbon fixation occurs in the mesophyll cell only. Dia hanya melibatkan sejenis sel saja untuk Calvin cycle berlaku untuk hasilkan sugar. Okay, mesophyll cell. Tapi kalau untuk C4, carbon fixation occur twice. First in the mesophyll cell, second in the bundle sheath cell. Ingat ya, bundle sheath cell is where Calvin cycle occur to produce sugar. Kalau C3 plant, first product is PGA, a 3 carbon compound, C3 acid. Kalau C4, oxaloacetate, C4 acid. Enzyme for C3 is RUBP carboxylase. Kalau C4 plant, enzyme is PEP carboxylase for first carbon fixation. Second carbon fixation is RUBP carboxylase ataupun Rubisco in the Calvin cycle. Okay. Kelas S62 ada tutorial dengan Sir Helmi kan? You all need to untuk keluar tak? Sekarang? Saya nak habiskan sikit lagi. Boleh?
Boleh.
Tapi kalau nak keluar, keluar je. Nanti saya upload recording. Alright? Saya nak habiskan sikit lagi. A few more slides. Okay, on C4 plant. Okay. So for chem plant. Okay, so chem plant is stand for crassulacean acid metabolism pathway. So dia punya nama dia berasal daripada family Crassulaceae. Okay, Crassulaceae, a stone crop family that lives in hot area, in desert. Okay, so these are other examples of plant that also can undergo chem pathway. Okay, so for chem plants, they live in really hot area. Okay, so what happen is that their stomata will close during the daylight. Okay, during daytime stomata close, so therefore carbon dioxide cannot enter into the plant tissue. So photosynthesis cannot occur kalau kalau carbon dioxide tutup pada waktu siang. Tapi apa yang adaptation untuk chem plant ni adalah, for chem plant dia punya adaptation adalah it opens stomata at night. Okay, stomata will open at night, so that is the time when carbon dioxide will be accumulated in the plant tissue. Okay, so carbon dioxide enter into the plant tissue when the stomata opens. So carbon dioxide is incorporated into organic acid that are stored in the vacuole. Dia punya reaction dia sama juga dengan C4 pathway. Reaction dia sama. Carbon dioxide combine dengan PEP jadi oxaloacetate jadi malate. Cuma malate akan convert jadi organic acid, which is malic acid, that is stored in the vacuole. Okay, that is what happened during night time. During daytime, when the stomata closed due to hot temperature and to reduce water loss, carbon dioxide is released from the organic acid. Ingat organic acid is malic acid. Okay, malic acid. So malic acid during daylight, kan, so dia akan convert jadi malate. Reaction dia ingat sama dengan C4, malate akan split into carbon dioxide and also pyruvate. Pyruvate generate back PEP. Carbon dioxide akan masuk the normal Calvin cycle to produce sugar. So basically the production of sugar will occur during daytime because Calvin cycle will occur during daytime. Okay. So kalau ini dia punya pathway. As I mentioned, chem pathway dia punya reaction dia sama juga dengan C4 pathway, cuma dia punya duration dia berbeza. Tapi location dia untuk chem pathway dia akan undergo carbon fixation twice. First carbon fixation during night time. Second carbon fixation, which is the Calvin cycle, occurs during daytime. Okay, cycle during daytime. So, but the location is still in the same cell, which is mesophyll cell. Okay, kalau C4 perbezaan dia adalah dari segi location. C4, first carbon fixation in mesophyll, second carbon fixation in the bundle sheath. Tapi kalau chem pathway, both carbon fixation first and second occurs in the same cell but duration is different. Nighttime and daytime. Okay. So itu je. Reaction dia sama. Carbon dioxide enter, stomata open at nighttime, enters into the leaf, combined with PEP, catalyzed by the enzyme PEP carboxylase, produces oxaloacetate, oxaloacetate into malate. Malate is stored in the vacuole as malic acid during nighttime. During daytime, when stomata closed, okay, carbon dioxide cannot enter. So the malic acid convert back into malate. Malate splits into carbon dioxide and pyruvate. Carbon dioxide will enter into the normal Krebs cycle to produce sugar and the reaction is catalyzed by enzyme Rubisco. Itu je perbezaan dia. So ini awak boleh baca, cuma ini just nak bagi tahu, okay, kalau chem pathway is similar to C4 pathway is that it undergo carbon fixation twice. Okay, twice. First by PEP, second by RUBP during Krebs cycle. Tetapi apa yang bezanya antara chem dengan C4 adalah kalau untuk chem, spatial separation. The location is different. First carbon fixation in the mesophyll cell. Second carbon fixation, which is the Krebs cycle, occurs in the bundle sheath cell. For chem plant, temporal separation. Perbezaan dari segi masa. First carbon fixation, nighttime. Second carbon fixation, Krebs cycle occurs during this daytime, but still it occurs in the same cell, which is mesophyll cell. Okay, so the last diagram here you see adalah gambarlah nak menunjukkan perbezaan antara C4 dengan chem. Okay, so C4 melibatkan dua sel, mesophyll, bundle sheath cell. Kalau chem melibatkan hanya satu sel, mesophyll cell, tapi perbezaan dia dari segi masa. Malam dan siang. Malam first carbon fixation, siang Krebs cycle. C4 first carbon fixation dalam mesophyll, second dalam bundle sheath cell, tapi semua dia berlaku masa daytime untuk C4 plant. Okay. So not chem, C4 and C3 plant all plant basically akan menggunakan Krebs cycle untuk hasilkan gula menggunakan carbon dioxide. So with that, habislah. Faham? Boleh?
Boleh Nadia.
Okay, clear? So, kita stop sampai sini dulu.